Good? Okay. All right, thanks very much. Thanks for the opportunity to be here. I'll be talking a little bit about genome graphs, and we'll start with motivation. So right now, you have a, sorry, this is taken from a longer talk. You have a situation where, yeah, we'll tell you someday about what the first two examples were.
You're going too fast, Dave.
You're going too fast, right? So right now, we have a reference genome that is just one representative of each of the human chromosomes taken arbitrarily from an individual. In some cases, it's a chimeric, where parts of it were taken from different individuals. That creates a bias, which is well known now, reference bias. So when you're looking at mapping millions and billions of reads, as we do every day, turns out there's a systematic bias. So if you're from an ethnicity that has different versions of regions of the genome, then your reads will not map against the standard reference as well as others who are from where that region is from the ethnicity that was used in the reference. We can't really tolerate this on a global standard. So many different groups are establishing their own reference genomes now. But if you think about this process, it's doomed to lead to some kind of crazy Tower of Babel. So we want to think about representing all of human genetic variation in one consistent reference data structure. And that's what this is about. And we're going to call it a genome graph. So it should include all common variants down to a certain level of frequency, say 1%, that occur in any of the ethnic groups that represent our human population. And we'll think of it as a graph. We can illustrate that in a minute. Now, this is of enormous research utility, but it is absolutely essential for the clinical application of genomics, which right now we're going through a process in which many genome technologies are now applying for approval by the FDA, by the clinical, the CLIA certification process, and in other countries the equivalent processes, so that they can be used to actually make clinical decisions. These have to then be a rigorous test which gives a well-defined answer. The problem with this is that most of these tests, when they're actually seeking a genetic variation, will produce the answer in one of many, many different types of forms. So there are a number of different databases and efforts that have been established to name the different human variants, and they, again, we have this Tower of Babel problem, especially because some of them focus on certain types of variants like single nucleotide substitutions or just substitutions of a few bases. Some of them focus on larger structural changes, and we have an enormous incompatibility, and this is right now the major problem holding us back from clinical application of genomics on a routine basis. It's still a research project because we haven't sorted this out. So what we need is some kind of a Rosetta Stone that will allow us to think about all of these different representations of different segregating human genomes that are moving around in the population, and all of the different different variations that occur on them. So a simple idea is to take individual chromosomes, which biologically, of course, are linear objects, and then merge the identical or common parts of them and leave the different parts as different paths through a graphical structure. So if you can imagine that the current reference genome is represented by this bold red line, that represents a long sequence of A's, C's, T's, and G's, perhaps through this region, the MHC, or major histocompatibility complex region, which holds on chromosome 6 about a 3 or so megabase region that has most of our immune genes in it. That one representation is actually a bad representation for this green chromosome, which deviates from it quite a bit. So if we zoom in onto this, going from the 3 megabase scale down to the lower scale, where now we're looking at essentially close to individual bases, we can see then the detailed process that you get as you compare genomes. Zooming in a little bit further, we see that at the individual base level, this red path had a typical segment for this genome, GCCAG. We see that there are alternate paths where the CA is replaced by a GT, or where the CC is replaced by a T, and so forth. So these are referenced as paths through this graph of DNA. And each of these positions, or in general, segments of positions that are useful in clinical applications, will have a unique machine-readable identifier along with the usual clinical names. But the idea is that this identifier would be permanent. The worst mistake we ever made was to start identifying these as positions that are offset from the beginning of the chromosome. I often say that if my PhD computer science student had designed that data structure and that way of representing places in the human genome, they would have gotten an F on their assignment. And the reason for that is that these locations are changing all the time as we learn more about the genome. Most of the genomes have regions that are unresolved, and this will remain so for quite a while, the centromeric regions, for example. But we are in this horrible situation that if in the current reference genome we realize that there's another base upstream for this, and we go from GRCH38 to 7 to 38, then the coordinates in absolute offsets of all of these change, utterly useless as a identifier. But this permanent identifier, maybe a UUID-like object, can remain for decades. So we need to name this variation with a local name that has a meaning within the graph that does not change as we learn more about human genetic variation, which we will inevitably do. So we add to this graph rather than subtracting from it. If there's something you really think that was bogus in the graph circa 2016, you can mark it as suspected or bogus, but you never really take anything away. So we are proposing to build this graph systematically. So I will look in this talk in detail about what we mean by the graph, what kind of graphs we built, and how we're evaluating them. And this work proceeds with a very simple definition. There were a lot of fancy definitions and discussion about how to represent these graphs, but the community, I would say, settled on a very, very simple definition. Rather than the pictures that I was showing where you were thinking about a data structure that would represent individual bases, for reasons of efficiency, we're allowing the objects in the node to contain entire strings of DNA. So the nodes of the graph will be strings of DNA in some default orientation. Like if you look at chromosome 1, it's oriented from the P arm to the Q arm by default. And then in between these segments of DNA, you have these things called joins. And the joins come off one end or the other. So this is not an abstract node, but it has effectively a 3' end and a 5' end, or a left and a right end to it. That means informally that these edges could be bidirected edges. If you represent this as a simple graph node, then the edge has to contain the information that says which end of the node you're attaching to. But whether you put that information in the node or the edge, by making two nodes out of this, effectively, we don't care. So conceptually, that's it. Now, DNA is great. It's like a railroad track that you can go either way on. And so you can have a connection from one end of this node. Normally a path would tend to go left to right through this structure, but we do not want to demand that it is a directed acyclic structure, because we do have cases like inversions that are possible changes in the DNA as represented here. So I could have the DNA ACG, ACGT, GG, GT, and blah, 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 like this. Or if this is inverted in some people, I could have the DNA that goes T, G, G, and then goes over here, and then starts traversing backwards through this by T, T, G, 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 and so forth, reading the reverse complement, and then picks up here and continues on. So it's important the double-stranded part of DNA is baked into the graph and its interpretation. Okay? So that's it. A genome graph is just a bunch of nodes connected by joins. Simple as that. Each node has a DNA sequence in a default orientation which tells what the ends are. Now, the project that I'll be talking about was sponsored by or organized by the Global Alliance for Genomics and Health. And a couple of words about that really quickly. It is an organization that is a non-governmental, not-for-profit organization founded by myself and 7 other individuals about 3 years ago. And its mission is simply to help the world share genomic data. And we're talking about sharing not in terms of privacy policies in this talk, which are a big part of the Global Alliance, but in terms about sharing genomic information, in particular variants, in a precise form and a durable form. All of the work at the Global Alliance is done on this GitHub site. If you're not familiar with it or you're interested in it, just go on the GitHub site. There are hundreds, approximately 430 contributors right now to this large open source project. All groups are welcome to participate. If you look at what happens in a day-to-day basis, and I get huge email threads from this, decisions are made all the time according to Apache Open Source Foundation rules. Basically, you make pull requests to modify the schemas or the code. If you get 3+1s from other people working on the code without any -1s, then the pull request is executed and the code marches forward. So the code gets developed. The leadership is totally determined by the amount of contribution. So the people who contribute are the people who organize the hackathons, the meetings, and make decisions about direction. And we have, of the 385 organizations in the Global Alliance, more than 120 of them are private companies. So this is a way in which private companies can be part of the standards making for the area that they work in. And these include lots of small companies, new DNA analysis and genomics companies that you've all heard of. There are dozens of them just in the Bay Area, plus big companies. Google is in there, Amazon is in there, Microsoft is in there. So we have a lot of work from all different types of companies. They all are collaborating on the interface. They all plan to compete on the implementation once the API is settled. We're anticipating, we're at API 0.6 at this point. We're anticipating a 1.0 API by the end of the year. So what about this genome graph idea? This is an idea that is in a second phase. So this will be in API 2.0, actually. But we are in parallel, as we work on the current API, developing the next generation. And we have a number of groups that have provided genome graphs in an agreed-upon schema and format. And they are now, you can actually interrogate them and use them through the Global Alliance servers and apply many evaluations. So people are building these 3 parts of it independently, but interoperably. The regions that we chose to look at are the BRCA1 gene, the BRCA2 gene. These are the most famous genes for breast cancer susceptibility. The SMA regions, this is a muscular atrophy region that's quite complicated in terms of its duplication and rearrangement structure. The KIR region, which is a killer cell immune associated region, also very complicated in terms of copy number, and the major histocompatibility complex region. We are starting to do the centromere, but I won't be talking about that. So here are the groups that participated in our first organized graph building and comparison, and the methods that they used. So how do you build a graph from DNA sequences? Basically, the idea is to take the DNA sequences and merge parts of them that you are going to treat as identical within your graph. So then you get a kind of merging and diverging railroad tracks graph from your DNA. Many people start then with a multiple sequence alignment, which suggests places which might be identical and in the corresponding positions to be merged. Other extremes, we had groups that just use a De Bruijn graph. So the nodes in the graph were all 63-mer. For the k equals 63-mer, it's every node is a 63-mer, and you have to adjust it because in the De Bruijn graph the nodes overlap a little bit. In our graph here they don't, but it's easy to transform it. So there are many different approaches as given on here. The last approach which I'll mention is this 1,000 genomes. This is built by Eric Garrison at the Sanger, and it simply takes the reference genome and adds an alternate path for every variant in the 1,000 genomes VCF file. So the 1,000 Genomes Project has documented many human variations. Each one you can think of, if you look in the VCF record, as aligning to the main primary path of GRCH38, then doing something different, and then rejoining. We call that a bubble sometimes. You put in a bubble for all the SNPs, essentially, and you get that. The Simon's Genome Diversity Project has produced a similar set, but with much more ethnic diversity globally, over 200 different ethnic populations, and so we did the same thing with that, actually with Eric. So now if you break down these graphs and you try to view them not at the DNA base level, but at a little higher level, they look something like this. So that 341 is 341 bases of DNA in here that I'm not going to show you explicitly. And this is a typical SNP. So we have one version of it that's the blue is usually representing the primary or GRCH30, our current reference. And this would represent a SNP that's present in the 1,000 genome set, for example. This is Benedict Patens, who's my co-speaker here. I may call on him for some of the details. So he's leading the project, and he also designed his own graph methodology called Cactus. And so this is one way of deciding. Essentially, this all comes down to deciding when to merge and when to keep things separate. And so algorithms for doing that are still, there are many different approaches, and we were testing them. Camel is another approach to do that, based on a methodology that I talked about here a while back, which was context-dependent sequence matching. And then this is a third method. So these graphs look pretty left to right, but if you look at the De Bruijn graph, especially at K31, it's very much not a left to right kind of thing. It's very much a hairball kind of spaghetti graph, as you can see in this depiction here. Now the De Bruijn graph with 63 is a little more like this, like the other graphs that we see that were built by multiple alignments. There's no alignment in this De Bruijn construction, obviously. You're just looking at the content of segments of DNA. So in BRCA2, you can see graphs that are made by just the BRCA data that we provided for this exercise. So everybody was encouraged to try to make a graph out of some initially completely independent sequences for BRCA. BRCA, the region that we chose for BRCA2 is about 85,000 bases. And so you can think about, one representation of it would be just one block. You can think about this as representing other variants of it in a very, very high level mechanism. For instance, this graph, SBG, by Curaverse, has a representation where they use very, very large blocks, and then you explicitly kind of tile across and try to get variation between those. So there will be an enormous amount of data in here that's very, very, that normally would be incredibly well aligned, and you might be tempted to merge. But then, of course, you lose phasing when you merge too much. And so this is an extreme trade-off of, I really don't want to merge. I want to keep a lot of big stuff separate. The tiling path that Curaverse used is represented in kind of a tiling array diagram where they will have large sequences that they will choose one of these large sequences. Even if there are subparts of them that match, they will choose separate paths for these and then make a kind of a switching decision at some point. So in the comparisons that I will give you shortly, there are some control graphs or default graphs. First off, there is a graph for BRCA1 that just represents that region of its chromosome in HGR38. So HGR38, just with its single chromosomes, is a graph. There's just one path in the graph. There are also the set that we gave everybody of fully sequenced different versions of the BRCA gene. And one thing you can do is not merge anything. So this is a graph. It just has 3 different ways to go all the way through the BRCA region. So this has the variation that you would get. And then there's also, for the ones that do the fancy merging, in particular, there's a whole class of these that said, fine, you gave us these examples of BRCA38, but I'm going to go to the 1000 Genomes Project and add in all the variants there as well. So you had to tell us if you were doing this, but you were allowed to go to other sources and make your graph even richer. And when you do that, you get these bumpy graphs. And in order to see what the bumpiness did, we have this control graph which simply takes all the bumps and shifts them over by 200 bases. So you have complete nonsense set of SNPs. But it controls for the topology of the graph. And the topology of the graph is identical, but it has nothing to do with biology. All right, so what do you get? Cactus, fun merge. This is on the KIR region, this killer cell region, and at zoomed way out, you get some interesting hairballs. Shows you the enormous structural variation that you can obtain when you start merging similar regions between genomes in this. And MHC, ouch. Okay. So there is a lot of complicated similarity throughout MHC, and it's easy to overmerge it. These are situations where things can be improved by heuristics. So one of the things you might ask is, what is the basic structure of these graphs? So every base in the graph has a degree, essentially, of how many other bases it's connected to. Now, if it's a base within one of those segments, that's 1, because you have to go linearly across the segment. But when you reach the end of one of these nodes, you may have 1 choice, 2 choice, 0 choice sometimes. So these are the average number of choices that you have after a base, including the ones, in the sequences. And you see it's very close to 1. And for some of these, we maybe get up to 1.03. And the different graphs have different branchiness. And these are the 5 different regions that are shown here. So the regions are branchier in some, and then the graphs are branchier than some. Compression is just asking you, if I add up all of the bases these are for graphs that were built from the training examples. If I add up all of the bases in these training sequences and compare that to the number of bases in the final graph, what have I got in terms of compression? So compression by a factor of 2 would have shrunk the graph in size a factor of 2. Most of the graphs are compressing. Occasionally some are decompressing. So that's an interesting phenomenon. We looked at another topological way to measure the linearity of this graph. So if you imagine, as most graphs, that we can lay out the graph by assigning positions of all the bases from left to right, so that then all of the arcs then are interpreted as going along a linear distance, then you can ask for any position in this topological sort or linear ordering of the bases of the graph, how many edges cross that? So in a very simple linear graph with just 1, it would always be one edge crossing this. But in this case, where we have two alternates, a SNP, then when we linearize this, we're going to put one of them on one side and one on the other, and so there will be two edges crossing these cut. So cut width is, at a particular cut, is how many edges cross that cut. And we're interested in the average cut width. Now, we topologically sorted the nodes. You might ask, did we sort them in an order that minimizes the average cut width? But we didn't do that because we looked at Dick Karp's paper, and it showed that basically that was NP complete, right? This goes back quite a ways, right? So we have a heuristic topological sort to do this. So if you look at the average cut width, these are just some calculations of this. There's two crossing here, two crossing here, two crossing here, one crossing here. And so we get an average cut width out of that. Okay. So this is the cut width calculations. And this was a big surprise because this is now a logarithmic scale base 10. So we had cut widths that were 100 times higher in some graphs versus the others, which is very interesting. All right, so there is a lot of variation in what people think are a sensible way of merging this data into a graph. You can rapidly get enormous cut width versus small cut width. Again, we have to try as many heuristic algorithms for ordering the nodes as possible to make sure this isn't an artifact of a bad ordering. It's normalized. Yeah, it shouldn't be less than 1. It's some kind of scale shift. The other measure that we did, so those are just kind of the topological features of the graph. What do they look like? How big? There's how big they are. That was the compression relative to the starting data, the branchiness, and the cut width. So the other thing we want to talk about is how well they represent the raw data that we're trying to capture. One simple measurement is just to look at the k-mers in the raw data and see if those k-mers are represented as little paths somewhere in the graph. Then we would say that the graph recalls that k-mer that it should. On the other hand, if you compress everything like crazy down to like De Broglie 2 or 3, then you're going to have paths for tons of k-mers that aren't really represented, even in a large data set like the 1,000 Genomes Project. So that would be a lack of precision. So precision, if you get hit on precision, that means you've got too many k-mers in your graph, more than are biologically plausible as common alleles. And lack of recall means that you're leaving out actual human k-mers that should be in your graph. Here is a plot of precision versus recall for the various graphs, and you can see we do quite well. So in terms of recall, most of the graphs are capturing on the MHC region 99% of the k-mers that they should be capturing, and at a very high precision over here. Now, some of them fall off as you go through this thing, but these are an example of the trade-offs. Now, the control graph is horrible in the sense that it creates a lot of k-mers that are garbage and misses the ones that it should. The primary sequence itself, so this is where we are now with GRCH38, is missing a huge amount. So it's very poor on recall, which is not surprising, right? It doesn't contain the variation that we want to capture. If you look at BRCA2, this tightens up a bit. So now we're at 0.996 or 7 up here for these graphs that are performing well, still at a very nice precision. Next.
What was K over the?
What was K? 20, I believe, right? Benedict? K equals 20 for this? Yeah, you can do we did these for various Ks. These are just sample graphs. So read mapping. Okay, a more realistic actual field test. We want to map reads to this graph. Everybody maps reads to the reference genome, but now you can map reads to the graph. Does that help? Major question. So what we did was took a bunch of 1,000 genomes reads that were BWA mem aligned. So they already had a canonical alignment to the reference genome, GRCH38. And we tried to map those to our graph using a algorithm that I won't go into the details of, but it's Eric Garrison's VG algorithm, which is smart enough to map to a graph instead of having to map to a linear sequence, which is all I'm going to say about it. This is one of the nicer results. Now what you're seeing here is the portion of those BWA mem reads that were actually uniquely mapped goes up from 80 to 90% within the graph. And this is the portion that were perfectly mapped. Now, there's a certain threshold of error in the data because you can't perfectly map if there's an actual mistake in the read. And the error rate is like 1% or a little bit more than 1% out of these 150, 120 base reads. And so your threshold is up here just a little ways. The noise is not going to prevent you from getting perfect mapping higher than a certain amount. And unique mapping, of course, is going to depend heavily on how much you collapse, right? If you under-collapse, you're going to map two different places. So what was beautiful about this, I think, is that you get more perfectly mapped and more uniquely mapped. So this is a win-win. It's not a trade-off. Graphs are just better. So we were very happy with this. This means that when you had a place to perfectly map it, you found it, and then that was the only place you mapped in a well-merged graph. So win-win. This very, very strong argument, I think, that graphs will be good. This is for BRCA2. Now, the numbers get higher, but the differences in performance get lower. BRCA2 is a much less challenging region. Yes?
Yeah. And your definition of unique mapping is the one that minimizes the error.
Yes. There's one unique place that minimizes the error. Okay, so now the last thing I'll talk about, and which loops back to our very first topic. I said the motivation for this is that so we can call variance in a way that is medically rigorous and that the authorities will certify as being rigorous enough to make a medical decision based on. So we need to be able to call variance in genomes very precisely. So we look at this general way of doing it. Again, this is in Eric Garrison's VG pipeline. All of those tools became a kind of common core for a lot of what the graph group did. You start out with a read alignment, and then you can look at a pileup at a certain region on that. And you may have a SNP, for example. You may have an A here and a C here. So you're going to get a pileup of different reads. And you will see to what extent those reads can match a path in this graph. Now that's interesting, and so based on that, you can call two variants here, one path that goes this way and one path that goes this way. But what's interesting is you may also have a place where there's another, where you cannot exactly match a set of reads which you think is real to either of these paths. And in that case, you have to do what we call an augmentation to the path. So our philosophy is that and we know every genome is going to contain novel variation that's not been seen before. So you have to handle novel variation. When you think you have novel variation, what you do is you take the canonical reference graph that you've built and you add another path to represent that. So therefore, you have an augmented graph for which every variant you're going to call is a path in your augmented graph. So you've added structure in order to name your variants, if you want to think about it that way. Okay, that's called an augmentation of the graph. And then from that, you make your calls. Now, how would you call this augmentation? You might have canonical names for all of these paths. This augmentation path would have to say, basically, I go along to here, and then I go to a new DNA sequence, and then I rejoin the graph here. So there is a canonical name for that. So if two people discovered the same augmentation independently, if it's an augmentation of the same base graph, they can recognize that they both discovered the new augmented variant. And in fact, this is how we envision that new variants would gradually be added to the genome, right? Because people are augmenting them like crazy, and finally they say, well, let's just put it in the reference. Everybody needs this augmentation. Okay, so you got the idea of augmentation. All right, so what we did was compute various heuristics and so forth, and output what was essentially a sample graph which only contains that part of the genome that's actually exercised by the sample. So going back to this picture, this might be part of a very, very large augmented graph. But now when you're looking at the particular DNA sample that you're trying to find a diploid genome for, you're only going to use a small part of that huge graph. This represents all of human variation. This represents the heterozygosity in the guy or gal that you're analyzing, right? So the sample graph is a small part of it that's actually used in the read mapping process. Okay, so I won't go into the details of how the calling algorithm works. Suffice to say, it is a very, very simple calling algorithm that was roughly illustrated on the previous slide. Yeah?
General question, this is all for haplotypes or genotypes?
So we are counting here basically genotype calls at this point. We're not talking about long-range phasing at this point. I'm going to get back to that at the end of the talk. But mainly, we're calling individual variants considered as point variants, and we're interested in whether they exist or not whether they're correctly phased with the neighboring variant. Answer enough? Okay.
David?
Yeah.
The population is so big that effectively every variant is in the population somewhere, right?
But not at 1%.
Okay, so you're confining yourself to some arbitrary threshold of...
Well, the community is going to have to choose a threshold, because obviously the graph gets too dense if you go crazy with this, right? So that's a practical choice. Most people chose reasonable thresholds, right? It looked at the 1,000 genomes data. They didn't include a variant that was only seen once in the entire 1,000 genomes kit. That's not how we built our graphs. Okay, so let's look at this. Now we're counting. The key term here is number of augmentations. So this is how many times in my new genome, which normally wasn't part of my training set, except for some of the graphs like SNP1000G, we tested it, on these particular genomes, and they actually were included in SNP1000G. So there was a little cheating on that one, but for many cases, the genomes that we tested on weren't. And so in order to interpret the genome NA19238, we will have to do a certain number of augmentations for stuff that we think is unique and wasn't in our graph. In particular, if you use, for this case, BRCA2, easy case, if you go through the primary genome reference, GRCH38 for BRCA, you're going to, 37 times, you're going to have to invent something that's not in that simple GRCH38 one chromosome. If you use how many examples do we give for BRCA, actual BRCA training?
The actual haplotype data that we provided, it's only 3.
3. Oh, okay. So if you look at the 3 different versions of BRCA in an unmerged graph, that trivial graph that doesn't merge anything, you're still going to have to make 13 augmentations to interpret your data. But if you look at these other cases, you can see that we make quite fewer, on average, augmentations. So the point is that when we're using these graphs, what you're looking for is usually already there if you've built a rich graph. And that's what allows perfect matching much more often. Here is more interesting statistics for a much harder region, MHC, where you have to make 4,500 augmentations in the primary structure. But some of these had remarkably fewer structures, fewer alternates that you had to add. So this shows a win almost to the point where you had the unmerged graph, at this case, with which would have burnt you very heavily in terms of multi-mapping, right? That totally would have screwed you if we're discounting multi-mapping. You can do as much multi-mapping as you want in this. But you're already getting down there with some of these other ones, and radically fewer than augmentations than you would have needed for if you just had GRCH38. This really, I think, strongly illustrates that a bunch of information can be packed in these graphs without detriment to the kind of key algorithms that we need. And here's now a comparison on the Illumina Platinum genomes where we have the state-of-the-art algorithm, essentially the GATK3 pipeline, and we're looking at 1- precision here to make it look like a rock plot versus recall. And this is accuracy on the SNPs. And so your GATK standard pipeline is this one in teal. And this graph that uses the 1,000 genomes baked into it is beating it soundly. So we are calling variance better than the GATK pipeline. How do the graphs look and how do they compare? I'll go through this really quickly. So you can compare two graphs. Again, from either a string comparison, do they contain the same 27 MERS in this case or whatever, or from a topology comparison. And this Jacquard distance is the usual measurement of the symmetric difference between 0 or normalized between 0 and 1. And you can see the various graphs and how they compare here. And this is a funny thing. If you just restrict it to the sample that's being used, they are very, very similar. Down here, the difference is really small. Sorry, this came off the list, but this is 10 to the minus big number. So on the samples, they're very different, whereas at whole graphs, they're quite large. So you can have a very large graph that looks different from this, but when you thread the sample through it and just look at the part of the graph that's left after threading, those two different graphs give virtually the same threading. Now, a threading of this through the graph, what does the Oxford group call it?
Chromotype.
Chromotype, okay. It's basically a partially phased interpretation of your data, right? Because you're just going to have two lines through this thing that are occasionally merged and occasionally split when it's a heterozygous position. So those two graphs look very similar even when the whole graphs don't. And then more precise thing, this was funny, this was fun. We did a topology comparison by the following reasoning. So here you have two graphs. These are very simple graphs. They just have these bases in them as individual nodes. So every node has one base in it. And you might have a graph AGGT, AGT, and then you could have an interpretation of the sequence AGT that forms a basis for comparing these graphs. So the overall idea is we take two graphs, we throw a lot of sequences, each of which has a path on them, and then we see whether, if we merge so that they take the same path, what we get. And in this case, you have two paths on this graph that are depicted here. I could skip the first G or I could skip the second G. And when you try to merge these to their common core, you get this graph, which is well defined. So there's a graph homology from here to here, and then there's another graph homology from here to here. And this is the largest homology image of these graphs under that path identification. So there's a nice kind of math behind this. And so you get this graph, which essentially is saying any number of G's here. So that is called the core graph. And so we compared arbitrary graphs by computing this maximum homological core graph. And these are the results that we got from that. So you can see, actually, that these graphs, this is the primary graph, the camel graph, the cactus graph, are quite similar. There's finally the scale in many cases to each other. And they're quite different from the De Bruyn's. So this is a measure of essentially, two graphs are similar if you merge them so that all the paths were still represented in all the paths of both of them were still represented, but you merged the minimum amount you needed to in order to achieve that. So if they're identical, the common core is the graph isomorphism between them, which is unfortunately also a hard computational problem, so that's why we use these path heuristics to compute it. But if the graphs are different in structure, then it forces you to merge out the differences down to a core that is essentially a common homological product, homology product of those two graphs. So anyhow, summary is that the graph reference has a lot of potential, but we do need more extensive evaluation. There is no one right algorithm that we found to build it. More data is better, that was clear. So the people who used a larger amount of human variation baked into the graph did better. You have an issue of how we're going to represent haplotype, which was Ron's thing. So there's a difficult trade-off here, because as soon as, if I have two different haplotype genomes, and so I have two SNPs here and here, and I merge the stuff in between them, I lose all haplotype information linking those two SNPs. So we are now, Benedict is now working on a very interesting new idea that would recover some of that haplotype information in an auxiliary data structure. Because you want to know for this graph, not just how often do I use every node, but you kind of want to know how often I use different paths that are multi-node paths. And that information can be cleverly encoded in an auxiliary structure, and that would be very exciting. So then these graphs come up another level in which they represent a richer source of human genetic variation than simply counting and structuring things. So we're very excited about that, and we want to measure rigorously whether that improves things like alignment and variation calling when we do that, community efforts, and so forth. So I want to thank the people that were mostly involved, primarily Benedict Payton, who led the whole project, and the students that he worked with here, the first four, Eric Garrison at the Sanger, many people at other institutions, including Gil McVean, and I want to thank the Simons Foundation for funding this thing. Thanks. Yes? How far?
The slide that has cutting.
Oh, width?
Further.
That's quite a ways back. Yeah, there we go.
So this one basically you represent the directed graph. Is it directed?
Undirected, actually. The edges do have different types, but they're bidirected, technically.
Oh, so each edge have each type, different types. So some.
Right.
Some can have both direction, the other one can have just one direction.
Yeah, so it turns out that most edges go from, let's just use left and right. Most edges connect the right side of one sequence to the left side of another. But there are these funny edges that I showed you in the reversal example that connect the left side of one block to the left side of another one, or the right side. So we call those reversing edges. So we actually count the cut width differently for reversing versus regular edges. Technicality, but basically, yeah.
And if there is a repeat, let's say there's some cycle.
Yes.
So if there's a repeat, then
There's a cycle, you're going to have a reversing, right? So with respect to this, we talk about the left and right edges, and then if there is, I didn't show you this, but yeah, if you have a regular, not a reversal, but you have a regular cycle in which you can go back to here and repeat this again and again and again, right? Then you must have what's called a back edge. And so this was the original problem from Dick's paper, feedback arc, right? So you could ask the question, when I linearly order these and I impose a direction, assume that the edges are, get rid of the edges that are 3' to 3', so the reversing edges are a special thing, and then just classify all the edges and direct them, because now they connect a 3' side and a 5' side, or a left side and a right side. So direct them, forcibly direct them this way, from the right side to the left side. If you have a cycle in your graph, a normal cycle in your graph, you cannot avoid having one of those directed edges that goes back. So that would be the feedback arc question. And then the natural computer science question is, what order of these minimizes the number of feedback arcs? And welcome to Dick's world, right? That's NP-Hart. So we use heuristics to order this. And so I'm just telling about cut width, but we also count how many feedbacks you have and how many reversing edges you have and how they are treated within this. The reversing edges, don't make a lot of sense within this thing, but the feedbacks do. Many of the graphs have 0 feedbacks because people like to build directed acyclic graphs. They get burned when they're trying to represent inversions and stuff because they can't, or they're trying to represent, if you have to represent a slippery piece of DNA that might be 13 copies, 14 copies, 15 copies, 17 copies, 20 copies, and a directed acyclic graph, you've got to choose to represent explicitly the different paths for the different number of copies, and then you will top out at some point. So maybe at 20 copies in your graph. Somebody comes along with a genome with 21 copies, there's no path in your graph for that. But if you put a loop, I can have as many as I want. But I'm going to have a feedback arc when I try to order it. Yes?
Have you thought of how this generalizes maybe to RNA as well? So where
Yes.
you might have alternative structures to begin with and all the variation on top of it?
This major motivation for this was better interpretation of RNA. So our view, and we have an RNA-seq task team led by Barbara Wold. Lior Pactor is working very intensively on it. He's got a great new Callisto, this new aligner, or a new RNA abundance estimator. But the key was that many people want to study the relationship between actual DNA variation and isoform-specific expression, and it's very hard to do that in the current format, right? You have just one reference chromosome not representing the other possibilities. So our view of RNA is very similar to this RNA analysis. It takes off from this. So we feel that everyone should start with the reference genome once we've referenced graph genome. So once we have a graph genome we all like that has most of the variation we care about in it, then you're analyzing Susie's DNA and RNA. So you're mapping Susie's DNA here, and possibly the RNA as well, and you find that you need 246 augmentations to this graph to fully represent Susie's DNA and RNA. So you make those augmentations. Now, and thereafter, whenever you're talking about RNA expression, there's an explicit path for that isoform in the graph, and you can distinguish that from the other isoforms. That allows you to connect the genetics with the RNA expression in a way that's much better than you can today. So this RNA is a huge argument for this kind of thing. Yes?
So leaving cancer alone for a minute, there are other complex genomes like T cells and B
Yes.
cells. So I wonder if you can comment on the scalability or representation of T
Yeah.
cell rearrangements and B cell lineages.
So, the T cell rearrangements, we probably would not see a particular T cell rearrangement at a frequency higher than 1%, so they would all be augmentations. So when you did somebody's genome, then you would say, but I sequence a bunch of T cells, then you're going to get a bunch of augmentations for the different T cells, and you can put some stats on there about the abundance of the T cell clone. But this is all about, this is an area where you would have to make possibly hundreds or thousands of augmentations in the same place.
So the graph will explode in those regions.
Yeah, and then it'll break our first tools. Then you'll have to fix the tools.
Maybe you should have one more question. Don't forget that there's another talk.
Yeah, sorry. Yes.
So a few years ago, we were looking at aligning reads to a reference that includes the variance, but we wanted to stick to a linear representation of text. So we have basically augmented the alphabet for SNPs to include like something that is represented with the 16-letter alphabet, like AC, so variations.
So you're gonna name a diploid position.
So for instance,
sorry, heterozygous position with a new character.
yeah, for exactly. And then
Okay, so you don't care about phasing then?
Exactly. So we
Okay.
use the phase information as
Right,
well.
okay.
And then for indels, so for more complicated branches, because we wanted to stay linear, we just basically padded each branch to the end of the reference. So I was wondering if you have considered something like this or a hybrid approach where snips, basically this would allow you to
They collapse, right.
bubbles.
Right, so in that approach, these things would collapse to a new symbol, which meant an AC heterozygote position.
The benefit of that was that we could still use BWT-based alignment on
Yeah.
the single. Yeah, okay.
Interesting idea, and I'd like to see a tool built on top of this that way. I still claim that the community should keep this richer information as the base information, but then somebody could write a clever app on top of it to squeeze it and then cheat and push it through more standard pipelines that way. You've hit on the key thing, is selling this to the community. You have to get over the, oh my God, we've got to recode our pipeline problem, right? So that's the issue. That's the inertia. So yeah, the better we can adapt tools, the better. Thank you.